Hello, we're going to go over the answers, or I'm going to go over them for this quiz. Number 1, which process can only occur in the presence of oxygen and produce the most ATP in the cell? Out of all the metabolisms, the answer would be aerobic respiration. Number 2, the final electron acceptor in aerobic respiration, meaning at the end of the electron transport chain in the membrane, in the cell membrane of bacteria, oxygen would be the final acceptor of electrons when they go into the electron transport chain from NADH. So remember, these were like the grandparents that picked up the electron kids, if that analogy helped you. Oxygen is the grandparents. In anaerobic respiration, which molecule can be the final electron acceptor? There are 2. So this one would be nitrate, E, or sulfate. And this was in the glycolysis video. And in your slides, I think, in your slides as well. Which process generates ATP through substrate level phosphorylation? So a substrate is like what the enzyme is acting on. So like glucose. And so the answer to this one would be anything with glycolysis, because that's where the substrate is being changed. In the Krebs cycle, the substrate is being changed, but it's not getting a phosphate on it. So phosphorylation means you're adding a phosphate, usually from ATP. So that's phosphorylation. So where do we add a phosphate from ATP to the substrate, like glucose? Where do we do that? In glycolysis. Okay, so which one has glycolysis? Fermentation has glycolysis. That means it changes glucose to pyruvate and then pyruvate to acids. Aerobic respiration has glycolysis because it makes pyruvate, and anaerobic respiration has glycolysis. Citric acid cycle is part of these 2, but on its own, it's not doing the substrate level phosphorylation. So it's not adding a phosphate to a carbon group that the enzyme, or that it's in the reaction, that's changing in the reaction. So that's not the answer for this one, because in the citric acid cycle, we're just making, well, we made 1 ATP, but we're not phosphorylating, adding a phosphate from ATP to the substrate. If you remember in the first step in glycolysis, the first step literally from glucose to glucose 6-phosphate, that was phosphorylating the substrate. So therefore, it has substrate level phosphorylation in glycolysis only. Okay. And then number 6. What is the ATP yield from stage 2 in glycolysis? So in stage 2, we are making 4 ATP. If you count all the stages, stage 1 and stage 2, it will be 2 net ATP, because you use 2 ATP in stage 1. If this part doesn't make sense, you definitely need to review the glycolysis video. Okay. Which of the following is most likely not to ferment sugars? Okay. Oh, we kind of didn't mention this, so I won't put this on the exam. But obligate aerobes are cells that need oxygen. And so if they need oxygen all the time, that means they do aerobic respiration. Aerobic respiration. Okay. All right, number 8. Which of the following is not a product of glycolysis in microbial cells? Well, would be carbon dioxide. And, oh, oops. Oh, I guess multiple things. Sorry about that. So the only product of glycolysis is pyruvate. So all of these are not products of just glycolysis. So all of these are not products. So that means the answer to this one is multiple answers. So this should have been mark all that apply. All right. The next one is number 9. So how does the proton gradient generated by the ETC power ATP synthesis? So protons, yes, are H+. So protons are H+ molecules, which are coming from NADH and FADH. And then they get separated from the electrons in their electron transport chain, right? And then so the protons are going to flow through ATP synthase, causing ATP synthase to turn, and that changes ADP to ATP. So the answer is C. What would happen, number 10, what would happen if ATP production, if oxygen was suddenly unavailable to a cell? Well, oxygen is required mainly for the electron transport chain. So definitely the ETC would stop, and the only ATP you would get is from glycolysis and maybe the Krebs cycle or the citric acid cycle. So the answer is B, because the ETC is where oxygen is needed, because that's where the oxygen picks up electrons. So this one is where it picks up electrons in the ETC. Okay. What is the overall purpose of electron carriers such as NADH and FADH? It's really to transport high energy electrons to the electron transport chain. So without this, electrons are very dangerous if they are like not carried by these very, very important electron carriers. For each turn of the citric acid cycle, basically one acetyl-CoA going into the cycle, how many NADH molecules are released? Would be 3, right? So make sure you look at the cycle. How many NADH are produced in just the link reaction? So this means when pyruvate is changed to acetyl-CoA, that is going to be 1 NADH. The number 14, during glycolysis, how many ATP molecules are produced per glucose molecule in stage 2? Oh, I kind of already had a question like this, but just like in a different way. So how many are produced for every glucose is 4 in stage 2. In stage 1, if this was stage 1, ATP is 0. ADP, right? We make 2 ADP because we change, we use ATP to make ADP because we transfer a phosphate from the ADP, from the ATP. So we don't make any ATP in stage 1. We actually use 2 ATP. In stage 2, we make 4. All right. Glycolysis produces molecules of NADH. Why must NAD plus be regenerated for glycolysis to continue? We kind of talked about this we did talk about this last week. It's because it's a coenzyme, right? So it changes, it recharges and uncharges. So enzyme basically is needed, NAD plus is needed to accept electrons from carbon 1 glyceraldehyde 3 phosphate. So without that, this needs to accept that H with its electron from carbon 1. If it's not able to do that, like if this is not available, then carbon 1 has to keep its electrons and glycolysis can't happen. So we definitely need NADH, otherwise the electrons can't move from the substrate to NAD, to the NAD. And the glycolysis can't continue without this accepting the hydrogen. Okay, which prokaryotic cell structure facilitates the direct transfer of plasmids between cells? That's the pilus, sometimes called the sex pilus, as we talked in class, because like this is like the mating of the bacteria. And that's it.